Hello students, welcome to your class of botany. Today we will be dealing with one very important topic that you struggle a lot, that is your replication of DNA. In the previous lecture we have already done structure of DNA, but today the most important one is replication of DNA. And in the simultaneous lectures we will be taking care of replication, then your transcription and then translation. Okay, so guys, before I start with this topic, that is replication of DNA, let me give you a small basic. Okay, this topic is very important again for your NEET and yes for your board's preparation as well. So guys, when we talk about your replication of DNA, of course, DNA will undergo replication. DNA, I'm going to use different pens so that it will be easy for you to understand. DNA will undergo replication, and of course replication, then it's gonna do transcription, and yes, during the transcription it will lead to the formation of mRNA, and then finally mRNA will undergo your translation process, which will lead to the formation of proteins. Okay. And this complete structure is actually known as your central dogma. Okay. So guys, please pay attention. When we talk about your replication of DNA, DNA is present inside the cell, which is present inside the nucleus. So the replication of DNA happens inside the nucleus. And inside the nucleus itself, the transcription is going to take place. Understand this thing, guys, one thing very clearly. DNA cannot go outside the nucleus. DNA itself cannot go outside the nucleus. So if it is in need of something very important, say for example some amino acids, some proteins, so what it's going to do, it will make an intermediator. So how it's going to happen? Replication of DNA, replication means it is the process by which a double-stranded DNA molecule is copied to give two identical DNA molecules. Okay. So when we talk about this, so now when they have two identical DNA molecules, from one DNA double-stranded I got two DNA identical copies. Inside that, once replication happens, transcription of getting mRNA happens inside the nucleus. Okay, and once it is done, mRNA will move out of the nucleus to the cytoplasm to do the process of translation by which proteins are made. Amino acid, 20 amino acids are there. And about these amino acids, you can actually go through your biomolecules chapter, in which all the detailed structure analysis of your biomolecules are there. Amino acids. So there are 20 amino acids. So generally the 20 amino acids, when they join together, they are peptide through peptide bonds. So they will be polypeptide. So the complete polypeptide will be known as your proteins. Okay? And this complete structure is known as your central dogma. So now let's move ahead and start with the lecture. So when we are talking about your replication of DNA, okay, I will try to explain you in a very, very simplified format. So now let's see here. When I'm talking about your replication of DNA, before the replication of DNA, it needs some preparation. So let's talk about those preparatory things. First, what I need to see during replication of DNA is the origin of replication. Origin of replication, which is actually known as ori. Guys, prokaryotic cell is a very small circular structure, whereas your eukaryotes are your linear structures. Okay, so generally for your genetic research or for your further studies, we use prokaryotes because prokaryotes are circular, and yes, of course, they have only one ori. one special origin of replication from where the replication initiates. Okay? So, ori, I'll write it down, in your prokaryotes, only one ori is there, and that one ori is also known as ori C. Again, prokaryotes have only one origin of replication, and that one origin of replication is known as ori C. Whereas in case of eukaryotes, they have many ori. That ori is also known as ori, but we cannot use many ori. What happens is that every time different set of origin of replication would be there. Then what about the origin of replication? The replication is very unique. So the replication initiation should start from one point, not for each and every point. Okay? So this is known as, first we need to see that what organism we are taking. So for any genetic research, of course we will be taking a unique one. So in this case we will be taking ori C, that is your prokaryotes. Okay? And now let's move ahead with your second step. I am telling you all the steps what you need to go through in order to do replication of DNA. The second step is, of course, very important, activation of nucleotides. Activation of nucleotides, which is also known as DNTPs. Deoxynucleotide triphosphatase or triphosphates. Why am I saying this? Because these nucleotides actually help in the polymerization process. Generally what happens inside the cell, we have deoxyguanosine monophosphate, deoxyadenosine monophosphate, ATGC, cytosine monophosphate, deoxythymosine monophosphate. So they need to get activated with the help of an enzyme known as phosphorylase, along with some energy. So the end product which I am going to get is your deoxyguanosine triphosphate, deoxyadenosine triphosphate, deoxycytosine triphosphate, deoxythymosine triphosphate. These are the ones which will help me for the DNA polymerization. Okay? Now, after this, I am going to write down the third step. Agreed, I got the ori, fine, I have got the ori and I have got all the TPs ready. That means I have converted all the monophosphate into triphosphate. Now what? Now everything is ready. Now we need to start with the replication of process. So the third would be initiation of replication. Initiation of replication. Okay? Now let's see how we do about this. So, say for example, this is a DNA molecule which is double helical structure. This double helical structure, let me write down in close one. This is your double helical structure. Okay? Now, there is an enzyme which is known as DNA helicase enzyme. There is an enzyme known as DNA helicase, which also can be known as rep protein. So, this DNA helicase enzyme this DNA helicase enzyme binds here and it will help in unwinding. That means it will open up, it will unwind the DNA structure. double-stranded DNA molecule. Guys, can you see here? If you can see. So generally, what is the orientation of your replication? When the replication starts, this is the orientation that we have. So of course, the replication is of course where? 5 prime to 3 prime end, and it's agreed? 5 prime to 3 prime end, 5 prime to 3 prime end. What I wanted you to understand is that whenever the replication process initiates, right now what is happening? Right now the DNA is unwinding, DNA need to get prepared so that polymerization occurs. Whenever there is DNA polymerization, it will always happen from 5 prime end to the 3 prime end of the DNA. Okay? So right now, hopefully you are able to understand that DNA helicase is the enzyme which will help in unwinding of the DNA molecule. Unwinding means opening up the DNA molecules. Okay? Now, but there is a problem also. And what is the problem? The DNA molecule which has opened up, which is opening up. Now what happens? Might be a possibility when they are going to open up, at the end, they might start to coil also. I'll show you one structure here. What if they start coiling again? You are opening up the pleats. But what is happening? At the end, they might start coiling up again. So at that point of time, what we need? Or you can just write it down. We need to add SSBP. What is SSBP? I'll tell you. This is your, guys, you know the orientation, so I'm not going to mention the orientation again and again here. Okay, I will just tell you where these proteins are being utilized. So when I talk about SSBP, I'm going to use SSBP here. What is the work of SSBP? I'll show it to you here. SSBP will help in stabilizing the unwinded DNA molecule. And the full form of SSBP is single stranded binding proteins. They are being placed here. So if these are the SSBP, guys. If these are the SSBP, what they are going to do? They will help in stabilizing. The DNA molecule and prevent from recoiling. Agreed, guys? So this is your DNA molecule. Of course, with the help of helicase enzyme, you opened it up. But once you are opening, and then at the end, the tail region, they might start coiling up. So in order not to let that thing happen, what you need to do, you need to add SSBP, single-stranded binding proteins. These proteins will stabilize these strands, and then recoiling will not happen. Now I'm happy. Helicase has acted. My SSBP is there, okay, and so SSBP has stabilized the strand. Now what is happening? Now when the SSBP is here stabilizing the DNA molecule, so what is happening? More and more helicase is working. So it is opening up. So generally what happens when you are opening up just like this, something aggregates here. Okay, just think about a rope. If you are opening the rope like this, you are just like this, so what happens? It will start coiling up, or it will start collecting here. In the next diagram, I'm showing it to you. One, two, three, four. Now the fifth step that we have, releasing tension of the coil. And what do we mean by tension? I will show you. See this? Now they are opening up because they are being stabilized, they will not recoil. So this is what is happening now. Can you see? The tension has developed here. What is tension? That means in order to open it up with the help of helicase enzyme, above, in the above region, what is happening? They are collecting at one point of place. If you can see, they have collected here. Now I need to release the tension for the proper replication to take place. So in order to do that, I want the DNA strand to be back like this. And yes, of course, SSBP is here. SSBP here, we should not forget this. Helicase is working. What I have done, I have added an enzyme here, and the name of this enzyme is topoisomerase enzyme. In eukaryotes, this enzyme is topoisomerase, but when we talk about prokaryotes, in prokaryotes, it is DNA gyrase. Okay. Again, so once what happens, once it is being open, so what happens at the upstream area, generally they start coiling up very, very densely. So what happens? A tension is developed. In order to release tension, we add an enzyme which is known as topoisomerase, which will release the tension. It will release the tension. Okay. And once it releases the tension, then of course the coil comes back to the normal state. Yes, one important thing. If it is happening in prokaryotic cell, instead of topoisomerase, it will be DNA gyrase. Topoisomerase is for eukaryotic cell. Okay? Everything is now placed, so everything has happened. Now let's see what we are supposed to do now. It is opening up, very nicely opening up. Okay, now isomerase is also there, everything is happening nicely. Now, initiation of polymerization. This is what we are supposed to do. We are supposed to make a copy. This is what is the essence of a DNA replication. But in order to do that, let's see what happens. But before this starts, this is the normal structure. I'll show you with the help of a structure. This is your DNA. This is your DNA which is still opening up because helicase is working. Let me write down the orientation. This is your 3' end, 5' end, 3', 5'. In order to start the polymerization process, we need to have DNA polymerase. DNA dependent DNA polymerase. We need to have DNA dependent DNA polymerase. But DNA dependent DNA polymerase cannot bind here because it needs a helper. So the helper is RNA primer. Let me first tell you what do I mean by polymerization. Polymerization means once something attaches, it will make a copy. That's a polymerization. Let me write it down what I have just told you. It occurs with the attachment of DNA dependent DNA polymerase. DNA dependent DNA polymerase. Of course, DNA dependent DNA polymerase is supposed to attach here and initiate the process. Okay? But DNA dependent DNA polymerase cannot add like that. It needs a helping hand. So the helping hand is done with the help of, it occurs with the attachment of DNA dependent DNA polymerase, but needs RNA primer for it. Okay, let me tell you what is RNA primer. I am telling you what is RNA primer. RNA primer is a small segment of DNA. RNA primer is a small segment of DNA which is synthesized by RNA primer. Who synthesizes it? Which is synthesized by DNA. Let me write it down with other colored pen. Synthesized by an enzyme called DNA primase. Remember guys, wherever then the suffix, in the suffix, wherever there is ase, maximum majority or the majority of the enzymes, they have suffix ase. Lipase, ligase, topoisomerase. So of course this is also an enzyme. Again, let me simplify it for you. This is what has happened. Everything is balanced. Now they have to start the polymerization process. That means they have to start making the copies of DNA, because this is what replication is all about. So right now we are at the last step of it. So in order to do that, to this strand, of course to this strand, because it's a DNA dependent DNA polymerase, so of course where it's going to attach itself, it's going to attach to the, where happens 5' to 3' end. Agreed? So now what happens? So it needs an attachment for the, at the 5' end to initiate the process, but it cannot do so. DNA dependent DNA polymerase cannot initiate your replication process. In order to do that, it needs RNA primer. And RNA primer can only recognize 3' end. RNA primer can only recognize 3' end. So what happens here? I'm going to attach RNA primer with a red pen. Say for example, RNA primer attaches here. And the orientation would be 5' to 3'. Agreed? And I told you that the DNA polymerization occurs from the 5' end onwards. So once the primer, this is your primer, RNA primer, which is of course a small segment of DNA, and it is synthesized by DNA primase enzyme. Now what happens when this RNA primer, this is RNA primer binds here, now then your DNA dependent DNA polymerase also comes and binds. This is your DNA polymerase. Before I explain further, let me tell you one more thing quickly. When I am talking about DNA polymerase, in prokaryotes are of 3 types. DNA polymerase in prokaryotes are of 3 types. DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. DNA polymerase 1, it is the one which also helps in the, these are the one which will help in the polymerization process, but they have some additional functions. DNA polymerase 1 is the one which will remove RNA primer. Whereas your DNA polymerase 2 is the one. DNA polymerase 2 and DNA polymerase 3, they both are fit. They are both fit for the polymerization process. But the problem is DNA polymerase can only polymerize 2, can only polymerize 50 nucleotides per minute, whereas they can do 2000 base pair per second. Because of this efficiency, DNA polymerase 3 is used. Now let's come back to the topic again. Okay, this is the one which we were doing. So now what happened? DNA polymerase 3 is going to bind here, and then it will start the process. Then DNA polymerization happens. This RNA primer will bind here also because it is supposed to bind. But I told you RNA primer has more affinity through 3' end, 2 3' end. There is no 3' end. So what happens? A complete segment of DNA will not attach, a fragment will attach. So it's going to attach like this. That is why you get the name. Again RNA polymerase 3 is going to bind here. RNA polymerase 3, they are going to bind. They will start the process, initiate the process. No problem. But here you will get continuous strand. And here you will get, which is continuous strand, that is why it is also known as leading strand. Here you will get discontinuous strand, which is also known as lagging strand. And these small fragments of newly synthesized DNA are known as, Okazaki fragments. So what you get is something like this. I am just going to draw it here. So what at the end you are going to get, I am just going to draw it here. At the end you will get template strand, agreed? And this is your newly synthesized strand, template strand, newly synthesized strands. These are the strands, the breakage that you get, the strands. These are the strands. Why? Because RNA primer could not get 3' end binding site, but they had to bind. Because once they bind, then only DNA polymerase is going to bind there. So at the end of the day, what you get after replication, you get one which has a complete continuous strand, the other one which will have discontinuous strand. So the discontinuous strand are joined by DNA ligase. And yes, I told you, this is what you get. This is what you get. And yes, I told you, if you remember, that still the primer is there. So now we need to get rid of primer. How to do that? Of course, DNA polymerase 1 will come, and the DNA polymerase 1 will get rid of RNA primer. And this is how we get leading strand and lagging strand. And these lagging strands, which are the newly synthesized DNA in the fragment form, also known as Okazaki fragments, which are later joined by DNA ligase. So this is what all your replication deals with. Hopefully this lecture have been like a bit of useful to you, and it will help you to clarify the topic. So if you have any doubts, what you can do, you can go back to the comment section and put your questions there. Okay, thank you so much for the lecture, guys.